Good morning, good afternoon, and good evening, everyone. Dear our respective evaluator, Dr. Wayan Sumardika, S.Ked M.Med.Ed, Ph.D. Today, SGD B4 will make a presentation with a title Pharmacogenomics. Okay, so here the member of SGD B4.
We are going to talk about the pharmacogenomics. There are three parts.
Okay, good morning, afternoon, and evening. My name is Sanet Malinda, and my last student number is 092. I will explain about the backgrounds of pharmacogenomics. Pharmacogenomics is a branch of pharmacology, which is the study of the drug's effect of genetic variation in organs on drug response. Pharmacogenomics also study the use of genomic science and technology in the creation, discovery, and development of drugs and their use in the diagnosis and therapy of disease. Scientists who are members of the Human Genome Project have announced the result of sequencing about 100,000 human genes on 26 June 2000 at the White House. Genes are very important factor for treatment. The definition of the gene itself is the smallest unit that encodes information about the inherited of a trait or many genes are called genomes. The genomes can decrease or increase protein that the body needs. A shift or change in both the compound and the abnormal amount of protein will produce abnormalities or disease. The difference in the pattern of protein provision or what is commonly called gene expression varies between individuals, and this results in difference between individuals. That is what causes a drug to be effective and safe for a disease that someone suffers from, but it can also be ineffective for other sufferers or even cause side effect or toxic effect in other sufferers, even though the disease is the same. In addition, this treatment also aims to diagnose a large number of types of disease that arise due to abnormal gene expression and early detection of a number of diseases by identifying the possible risks of certain diseases such as cancer, heart disease, and hemophilia.
Hello, my name is Anastasia Gurutroslin. My student ID number is 089. This is the problem formulation of pharmacogenomics. The first one is what is the definition of pharmacogenomics. The second one is how does the mechanism of pharmacogenomics work. The third one is how is the metabolism of pharmacogenomics drugs. The fourth one is how does pharmacogenomics work. The fifth one is what is the application of pharmacogenomics. And the last one is what is the applications barriers of pharmacogenomics. And this is the proposal on benefit of pharmacogenomics. The first one is definition of pharmacogenomics. The second one is the mechanism of pharmacogenomics work. The third one is metabolism of pharmacogenomics drugs. The fourth one is pharmacogenomics work. The fifth one is application of pharmacogenomics. And the last one is application barriers of pharmacogenomics.
I will continue the presentation. My name is Anggia Manda Tranadevi with last 3-digit numbers. Student ID numbers is 088. Many drugs thought will fit all people, but actually that is not the reality. Genetics heterogeneity in every individual can lead to different outcomes of the drugs that they consume. Human body responds to drug influenced by some factors such as physiological factors, environmental factors, and also pharmacological factors. The diversity of drugs response that people give make it hard to predict the effectiveness of a drug for specific patients. So, pharmacogenomics is used to answer this problem. What is pharmacogenomics? Pharmacogenomics is a field of study that focuses on how variation in genes affect individual body response to pharmacological treatments. This field of study consists about the development of drugs' effective doses and safe medication designed according to an individual patient genetic. Pharmacogenomics is a combination of words pharmacology, which is a branch of science studying about drugs, and genomics, which is a branch of science studying about genes. Even though pharmacogenetics and pharmacogenomics use consecutively, pharmacogenomics is different from pharmacogenetics. Pharmacogenetics studies on variation of drug response that influenced by a single gene. Meanwhile, pharmacogenomics studies on influence of the entire gene spectrum. Pharmacogenomics is an essential field of study because it can help to improve drug efficiency and reduce adverse drug reaction.
My name is Azamir Salfatila with student ID number 094. There are various different ways in which our biology can affect our body's response to drugs. The two main interactions between drugs and the body are known as pharmacokinetics and pharmacodynamics, and both can be influenced by genomic factors. The first one is pharmacokinetics interaction. When we take a drug, The drug will travel through the body to its target and then leave the body. Pharmacokinetic genes facilitate the first and the last of these processes through ADME processes, how it is absorbed, distributed, metabolized, and excreted by the body. These parameters decide how fast the drug reaches its target and how long its action can last. One important parameter, bioavailability, involves the fraction of the dose of the drug that ends in systemic circulation. Intravenous delivery will provide 100% bioavailability, while an orally ingested tablet or capsule may be incompletely absorbed by gastrointestinal tract or metabolized before it reaches systemic circulation. The genomic alteration may impact the bioavailability of the drug at its target sites. A good example is cytokine CYP2D6, a protein which plays a role in the body metabolism of many drugs. Variation of a CYP2D6 gene determine whether an individual is a fast or slow metabolizer of certain medication. If an individual is a fast metabolizer of CYP2D6 sensitive medication, the drug will usually be less effective because it is broken down and eliminated from the body more quickly. Conversely, slow metabolizer may be more susceptible to side effect because the drug concentration builds up in the body.
Okay, my name is Alamanda Chandra and my 3-digit student ID number is 086. I'll continue the presentation. As what my friend has explained before about pharmacokinetics, or in short, metabolism of drugs, now I'll explain about pharmacodynamics, which the drug has its own effect to the body, excluding how the metabolism occur inside our body. Drugs will give effect in our body when it arrives on the site of action. The interaction between those two can lead to therapeutic or toxic effect. And it has the correlation between gene variant that people have. For example, carbamazepine has therapeutic effect for epilepsy, but gene variant in HLA-B1502 has hypersensitivity that affects skin, and the worst case leads to Stevens-Johnson syndrome and death. Last thing I want to cover, the double whammy. It means that two things can happen at the same time. Genetic variation can cause pharmacodynamics interaction and pharmacokinetic pathway. For example, warfarin gene variation has those two effects. As for the pharmacokinetic effect by CYP2C9 and pharmacodynamic variation affect the protein involved in blood clotting by gene VKORC1.
My name is Theodore Manzinauban. Last digit student ID number is 093. So today I'm going to explain about the drugs metabolism. Drugs metabolism is needed in our body so the drugs can be absorbed in our body. First, the principal organ that we use to metabolize drugs is liver. Even though every tissues have the ability to metabolize drugs, but liver is the principal organ. Based on Jennifer Lee 2020 text on drugs metabolism, drugs can be metabolized by oxidation, reduction, hydrolysis, hydration, conjugation, condensation, or isomerations. So, drug metabolism could react differently within patients. There are patients who have rapidly metabolized drugs there are patients with slow drugs metabolism that also affected how drugs metabolize in our body. The patient who have the rapidly metabolized drugs, they have a smaller chance to have a toxic effect for usual doses. But if the slow metabolism, they are regularly to have the toxic effect for usual amount of doses for normal patients. So after drugs are eaten or oral administrations and absorbed by small intestine and transport to portal system in the liver, there are two types of phases that are needed to metabolize drugs. The first phase is called the microsomal mixed function oxidase system. So what the mainly thing in the first phase do, they do the oxidation and reduction process by producing flavoprotein. The second phase that are used which is use chemical groups with conjugation reaction. Phase 2 is faster than the first phase that need P450 catalyzed reaction that makes drugs effective rapidly accelerating drugs biotransformation. I'm going to explain a little bit about the drugs metabolism toxicity and drugs metabolism factors. The toxicity reaction might occur when our body or resources get exhausted, might be toxic pathway such as carcinogenesis. So what are factors that are used to our drugs metabolism? Maybe such as the individual differences, genetics factors, dietary, environment, and age and sex. That factors also have also affected the drugs metabolism in our body.
Hello, my name is Christian Salomo Paracian Sinaga with last 3-digit student ID number is 096. So I will tell you about how pharmacogenomics works. Cytochrome P452D6 is associated with the digestion of up to 1/4 of all medication utilized clinically, counting overwhelmingly essential mixes and, for example, beta blockers, antidepressants, antipsychotics, and narcotic analgesics. Among these CYP compounds, CYP2D6 is answerable for digestion of about 20% of clinically utilized medications. Like other polymorphic proteins, four clinically characterized metabolic aggregates, i.e. PMS, IMS, EMS, and UMs, are utilized to foresee remedial and unfavorable reactions following the organization of CYP2D6 substrates.
My name is Putra Dinarshati Setiawan with last 3-digit student ID number 097. Now I will explain about the application of pharmacogenomics, but I will explain just the first point and the second point. So in this time, the advancement of pharmacogenomics is fast in the medical world, and in its application, there must in any case be refreshes and further exploration to be ground with the goal that its usage can run optimally. Pharmacogenomics, or PGX, aims to decentralize the genetic basis for inter-individual differences in drug response. Pharmacogenomics enables health workers to use the patient genomic information to make an important decision regarding the type of drug or dosage of the drug to be administered to the patient according to their suitability and needs. And pharmacogenicity is also valuable for improving health outcomes, reducing healthcare costs, and avoiding adverse drug reaction. The PGX, or pharmacogenomics, approach has been applied to numerous anti-cancer medication with an end goal to identify relevant inherited or acquired genetic variation so as to predict a patient response to target chemotherapy and treatment. And the study has been studied quite effectively for long-term and outpatient inpatient. Until now, there have been many advances in the field of pharmacogenetics cancers, which arise from the encouragement of the latest and revolutionary technological advance, as well as their application to decentralize the genetic base for inter-individual drug response and the development of biomarker capable of personalizing and specifying treatment.
My name is Yosefina Graslataji, and my student ID number is 087. Okay, I will explain the last 2 points about the application of pharmacogenomics. There are things to note in application of pharmacogenomics, namely the label of medicinal product. Drug products label need to be considered well for the safe and effective use of medicinal products for our patients. The effect of pharmacogenomic testing is absolutely amplified when executed over the populace, and also the pharmacogenomics variations that can be applied any place, and test result can be utilized forever, permitting clinical staff to utilize these outcomes preemptively. And the last, it is trusted that pharmacogenomic will decrease adverse drug reactions, or ADR, and related to some medical care issues.
Hello, my name is Felix. My last 3-digit of student ID is 099. So I'm going to discuss about the implementation barriers of pharmacogenomics. As we know, healthcare workers use patient genetic information to create a personalized drug that are safer and more effective. However, in implementing pharmacogenomics, there are still some flaws that require extra attention, such as information technologies, education, cost insurance coverage, and ethical issues. First, I'm going to explain about the information technologies. In clinical aspect, the application of pharmacogenomics collaborates with the information technologies such as clinical decision support system or CDS and big data technology to provide efficient treatments. A CDS system is a computer-based program which function to assist physician or medical personnel in making clinical decisions. However, the increasing knowledge or information from the genetic research causes problems for hospital in collecting and storing data, deciding the structure of CDS system and optimizing the user interference. Second, the education. Lack of pharmacogenomics knowledge and course training on both patient and physicians are a major issue problem in applying pharmacogenomics in the healthcare system. This leads to many patients and physicians that can consider the importance of pharmacogenomics as an alternative way or method to improve the treatment.
Hello, my name is Jupin Lufianto with last student ID 091. So I will continue about costs and insurance and also ethical issue. So for genetic testing, it has high cost. The cost is depending on the nature and complexity of the test. The cost increases is more than one test is necessary or if multiple family members must be tested to obtain a meaningful result. Only few types of tests are offered by the insurance policy. Furthermore, insurance approval process could involve multiple steps, requirements, and a long period of waiting. About ethical issue, pharmacogenetic tests involve collecting and storing sensitive information on patient genetic information. So it triggers questions regarding to privacy. Patients must be well informed about how their genetic information will be handled, processed, utilized, stored, and protected. The patient must be informed about this, who will have access to their genetic information.
Okay, thank you. My name is Putu Yudhistira Kawardana with 3 last digit student ID number 098. I will giving the conclusion from this presentation. Pharmacogenomics is field of science that including both investigation of pharmacokinetic and pharmacodynamic in the utilization of genomic data with an end goal to make drug treatment more explicit to every person to diminish the harmful impact of medication to abase and increment the impact of medication treatment, however much as could be expected. Pharmacogenomics can be supposed to be a generally useful field of science, but however, this field of science has gained fast ground in its turn of event. Pharmacogenomics has applicated in the clinical domain by helping specialists and well-being laborer in settling our treatment decisions that are more explicit and obviously betterly affect patients. Despite the fact that pharmacogenomics offer numerous points of interest, pharmacogenomics additionally still face a few impediments. For example, medical clinic that experience issues deciding clinical decision support or CTS. There are as yet numerous individuals who don't comprehend and well-being labeler who are not all that very much prepared, cause issues and moral issues in execution of pharmacogenomics. So later on, pharmacogenomics is normal will turn into the main and broadest field of clinical genome as far as clinical execution and give a bit of detail to singular patient around this world. Next slide. Okay, I think that's the end of this presentation. We are sorry for our mistake and thank you.